Next we have Ksenia Blinova from DARS to talk about the FDA progress and collaborative opportunities. Ksenia.
Good afternoon, everyone. I know biologically you're post-lunch food coma. I promise to speak in soft and monotonous voice so I don't interrupt anything. You can come back in about 20 minutes when I show my summary slide and it will be all cut up. Let's begin. So my name is Ksenia Blinova. I'm Associate Director at the Division of Applied Regulatory Science at the FDA CDER Center for Drugs. Today, I'm excited to share FDA's progress in translating NAMS innovation into regulatory practice and discuss collaborative opportunities moving forward.
There's a button on the side.
Side.
Turn down. Yep.
Okay.
Sorry about that.
Thank you. I'm sorry, it was so exciting. I didn't plan for it. This is disclaimer. Those are my views and not represent FDA policies. So let me start by highlighting some significant milestones that happened since our last CIVM workshop. In April 2025, FDA published a roadmap to phase out animal testing requirements for monoclonal antibodies and other drugs. This is a major step forward in our commitment to modernization. In May 2025, the Government Accountability Office published their report on human organ on a chip technologies, recognizing the benefits this system offers over animal testing, while also acknowledging the challenges and currently limit wide adoption. In June 2025, CDER established the CDER NAMS Coordinating Committee to serve as a central point of contact for FDA and external stakeholders, ensuring alignment, transparency for NAMS space. You can reach us at this email below. And then in July 2025, ISTAND, the Innovative Science and Technology Approaches for New Drugs program, became a permanent qualification program. So everyone here is probably familiar with how the ISTAND program fits within the FDA development tool programs and more specifically within the drug development program, DDT. On the bottom, you see the three-step qualification process: letter of intent, qualification plan, full qualification package. But one important point that I want to make here is about the appropriate use of these pathways. We're seeing some confusion around when ISTAND application is the right approach. So let me try to clarify the distinction. ISTAND is appropriate when you are developing a NAM for regulatory decision-making in an IND agnostic manner. What do I mean by that? For example, if you want to use a specific assay to waive an animal study across multiple drug development programs, not just for one particular compound or one specific IND, then ISTAND qualification is the right pathway. The goal here is to establish that the NAM can be broadly applied to regulatory decision-making. On the other hand, if you are developing a NAM primarily for internal drug development planning to guide your company's development decisions and strategy, that doesn't require regulatory input and it's not appropriate for ISTAND qualification. However, we're still interested in hearing about those innovations. You can engage with us through other mechanisms such as the Critical Path Innovation Meetings or the Fit for Purpose program. Additionally, and this is also important, you can absolutely come in and discuss the specific NAM for a given context of use within the specific development program. For instance, if you want to use a particular organ on a chip model to address a specific question for a particular drug candidate in your IND, that conversation can and should happen through the usual IND channels with your review division, not through ISTAND. So with that in mind, let me show you some of the submissions most recently accepted by the ISTAND. You may remember at the last CIVM workshop, we celebrated the first MPS letter of intent accepted in the first stage of the program by Emulate. Now we have like many more. We have MPS or CIVM-based tools for drug candidates, those in liver disease, another multi-site tool for DILI from 3R's collaborative, human kidney chip for nephrotoxicity, reproductive toxicity, iPS cardiomyocyte assay for cardiovascular repolarization risk assessment. There is also a number of new submissions using AI, ML technologies in the mix. So another point here is the range of requesters from industry partners like AstraZeneca, CROs like Charles River, to academic universities, platform developers, and now from collaborative nonprofits that may represent tools developed by multiple developers, but for a similar context of use. This really demonstrates the broad stakeholder engagement that we are seeing. So all these submissions, along with the FDA acceptance letters, are available for public review on our FORCE platform. It's a searchable database that you can access through the link on the bottom. So this pie chart gives you a snapshot of the ISTAND portfolio as of late February. Looking at the breakdown by technology type, MPS and CIVM technologies represent 40% of submissions, demonstrating the strong interest and innovation in this space. AI and ML approaches account for 31% of submissions, reflecting the growing role of computational methods in drug development. So beyond the 16 accepted letters of intent, we have accepted 2 qualification plans, and now the first full qualification package is in the pipeline. So strong progress since our last meeting, and I like to think that our in-depth discussions on, at this last meeting a year and a half ago on how to craft a good context of use is part of that success. This graph shows the trajectory of qualification submissions over time. You can see clear growth from 1-2 submissions per year in the beginning of the pilot ISTAND program to about 40 submissions in 2025. And we expect this number only to grow. So, slightly different topic. One of the critical ways FDA supports the translation of NAMS innovation into the regulatory practice is by building internal hands-on expertise with these technologies. This is where our Division of Applied Regulatory Science, plays a key role. DAR's mission is to engage stakeholders in mission-critical laboratory, computational, and clinical applied research to inform regulatory decision-making. Our vision is to move new science into the FDA regulatory process and address emergent regulatory and public health questions. What makes DARS unique is our multidisciplinary approach, available in one single unit. We have laboratory capabilities, computational science expertise, and clinical research capacity. Within DARS, our complex in vitro models laboratory, the CIVM lab led by Dr. Pfister, who is here today. So my point, I guess, is that we don't just review submissions, we actively work with these platforms ourselves. Our portfolio spans the full spectrum of NAM maturity. On one end, we work with more mature systems where validation studies may have been conducted, best practice recommendations have been developed, and the assays may already be listed as an alternative approach in FDA or ICH guidance documents. On the other end, we are evaluating models that are still in the infancy but address critical unmet need, and where we're hoping to be ahead of the curve, so when a cutting-edge science makes it into the regulatory submission, we are ready to review it. So let me now walk you through several examples. I'll start with more mature NAMs. This is first cardiac NAM that is really rooted in SIPA, the Comprehensive In Vitro Pro-Arrhythmia Assay Initiative. Represents a paradigm shift from reliance on traditional clinical thorough QT studies to a mechanistic assessment of drugs' propensity to induce cardiac risk. To assess the potential of human-induced pluripotent stem cell-derived cardiomyocytes to predict pro-arrhythmic risk, multi-site study of 28 drugs with known clinical records was conducted by 10 laboratories using 2 different commercially available IPS cell lines and 5 different electrophysiology platforms. The results were pretty impressive. The IPS cardiomyocyte assay categorized drugs into high and intermediate versus low proarrhythmic risk groups with 87% probability. Since this study was published in 2018, FDA received more than 100 of IND submissions where this assay was used. So, and in our most recent effort, we analyzed this real-world data, the data from regulatory submissions submitted to the FDA to compare this IPS cardiomyocyte assay to the standard non-clinical assessments such as HER-Block and in vivo animal QT studies, as well as with the clinical golden truth QT prolongation information when this data was available. So this concordance analysis revealed that this cardiac in vitro NAM is a promising alternative to animal testing for QT prolongation assessment. You can see the specific values for the assay. You're looking at the wrong slide. This is significant because now you can see sensitivity specificity of IPS assay versus animal data. And you see it's very good compared even to animal and single hERG assay. So it's really give us hope that we can reduce animal use aligning with our roadmap. So to facilitate consistent and efficient review of this new IPS cardiomyocyte assay, we were developing a teaching tool for our pharm tox reviewers by developing this mock submission on the left that mimics actual IND. The tool helps ensure that all required information is present in study reports and outlines how to interpret this data in proarrhythmic risk assessment context. We also developed the TDP risk assessment categorization or track calculator, an open source web-based tool for quick evaluation of Torsades point risk from in vitro IPS cardiomyocyte models. The calculator takes several key parameters from in vitro studies, such as, for example, drug-induced action potential prolongation, and calculates the probability of the drug to be in high or low proarrhythmic risk category. This tool streamlines TDP risk estimation for routine regulatory use. The software will soon be publicly available, promoting transparency and accessibility. In this pilot study, we have applied this in vitro assay to the assessment of proarrhythmic risk of drug combinations for the first time. A critical real-world scenario. So this case study tested moxifloxacin, a drug known to prolong repolarization, in combination with cabetastat, which shortens repolarization. Our results show that cabetastat partially mitigated the repolarization prolongation caused by moxifloxacin. This demonstrates that the assay can be used to understand the net electrophysiological effects of drug combinations prior to clinical trials, providing valuable safety information early in development. Moving to more complex models and lower throughput, we're working with human cardiac microphysiological system. This organ-on-chip platform mimics human cardiac tissue with integrated media inlet and outlet for nutrient and drug exchange. We evaluated this system with vanoxarine. Vanoxarine was a drug that was reported to induce cardiac adverse events, including Torsades points, and in phase 3 clinical trial. QT prolongation or proarrhythmia events were not detected early in non-clinical animal models or in phase 1-2 of clinical trials. Our IPS cardiomyocyte-based MPS model detected delayed repolarization and irregular heart rhythms these traditional models may have missed. Let me now move to neural NAMs. So given the ongoing opioid epidemic, we developed an IPS-derived neural co-culture assay using the ME platform to assess CNS opioid interactions in combination of these cells. We plated neurons and astrocytes on the microelectrode array plates, recorded baseline activity, and then we applied the opioid agonist and then naloxone. We analyzed multiple output metrics, including individual spikes, bursts, and network bursts. Our analysis showed that neural co-culture responding to the opioid agonist was suppressed activity, then reverting toward baseline through application of naloxone. So this early-stage feasibility study provides a foundation for non-clinical evaluation of opioid agonists and antagonists on human neuronal electrophysiology activity that has potential to support safety and efficacy assessment. We extended this work to clinically relevant combinations. Fentanyl significantly reduced network burst activity while co-administered with naloxone reduced network burst activity while was reversed with opioid-induced network suppression when naloxone was administered. We also work with respiratory NAMS. Here we'll be evaluating a human lung MPS for assessing apparent permeability of inhaled drugs. The model includes relevant cell types, ciliated cells, basal cells, goblet cells, producing mucin, and endothelial cells arranged in a physiologically relevant architecture with media flow, immunofluorescence. That's not me who's doing this. Somebody is clicking my slides, so I have to go back. Okay, we're on the right side right now. So our immunofluorescence characterization confirms the presence of proper localization of these cell types with tight junctions as indicated by staining. This project establishes quality control criteria for the small airway MPS and is consistent with the 3 R's principles, as the ex vivo rat lung is often used to assess inhaled drug permeability. This human relevant alternative has the potential to reduce animal use while providing more clinically and human relative data. This one is about lung MPS for predicting anti-infective drugs penetration, which is currently assessed in quite invasive phase 1 studies, clinical studies using bronchoscopy. We are comparing lung MPS data with existing clinical data to evaluate whether MPS could serve as an alternative or supplementary approach. Finally, let me discuss an emerging area, blood-milk barrier NAM. Breastfeeding is vital for infant health, but knowledge about medication safety during lactation is extremely limited due to the low enrollment of breastfeeding mothers into clinical trials. This lack of information is risky for both the infant but also for the mother, who may stop taking the critical medication, mental health drug, for example, in fear that it can harm the baby, so her own health could be at risk. So to close this knowledge gap, we are evaluating an MPS model that mimics the human blood-milk barrier with human memory endothelial and epithelial cells that mimic human memory tissue structure and function. The next step would be to test drug transfer across the barrier and compare it with the drug content in the human milk obtained from our partners at the existing milk biobank. If successful, this approach could be a life changer in assessment of medication use during breastfeeding. So if I made you curious about any of these research projects, please find me during the break. I'll be happy to connect you with the lead scientists on these projects who are here today. And I'm going to complete with my summary slides. So first, FDA is committed to modernization. We are committed to phasing out animal studies where scientifically valid alternatives exist, advancing more predictive human-relevant models. Second, we have coordinated leadership in NAMS. CDER has established the CDER NAMS Coordinating Committee to serve as a central point of contact for FDA internally, but also for external stakeholders. Third, we seen growing momentum in ISTAND. Uptake continues to increase, with approximately 40% of submissions focused on MPS and CIVM technologies, demonstrating strong innovation in this space. Workshops like this are critical to accelerating scientific exchange and regulatory readiness. And finally, DAR's regulatory science initiative supports CDER efforts in NAMS translation, and we welcome collaborations to generate the evidence needed to support qualification, adoption, and implementation of alternative methods. Thank you.